works related with photosynthesis, not deeply in light perception, but photosynthesis is regulated by many different stress conditions. For example, too much energy to highlight, or low temperature, or in some cases, in the desert, no water, or some nutrient is not available. So I'd like to introduce our work about the photosynthetic carbon concentrating mechanism, which support CO2 fixation. Of course, the most important thing, which organism should we use as a model? Because we work with poplar or some trees, it's a long time to get the results. So my friend is working bamboo, but it's flowering time is 10 years later, everything years later, or like that. So today's title is called calcium binding protein Cas and calcium regulate the expression of ABC type plasma membrane bicarbonate transport in chlamydomonas. This work is done mainly by the Chinese student. He spent 3 years and got a PhD, and right now working in postdoc in Princeton University with a high salary. So he's right now very happy, and I'm really glad he was able to PhD in this work, not only by himself, but many students and assistant professors helped him. And anyway, so let's start the introduction of the model organism I'm working on. This is what is we call clammy, but it is really single cell, looks like yeast, but green. So, and moving, that means the animal with flagella, and the green means they form photosynthesis. So they fix carbon dioxide for the component of any kind of biological materials by themselves. And also, they meet each other, female and male. Many other workers, researchers in medical school is focusing on how the mating occurred and how the flagella work. And of course, many people is human need cilia and flagella is very, very important. For example, is your heart is right side or left side? My friend has a right side. He has a little bit problem because of the flagella moving. So you know how the flagella is important. Right side and left side determination angle is determined by flagella streaming during development of our body. So this organism is used many different aspects. So I'm focusing on photosynthetic carbon concentrating mechanism. Because carbon is, you are eating everything, you are eating the material produced by photosynthesis. So photosynthesis is the most basic energy support and using light energy. So this schemes show remind you how light energy is perceived by photosynthetic right reactions centers, and water is splitted and oxygen is evolved. But by using energy, the ATP and the NADPH like redox energy compound is used for fixed carbon, and also sometimes reduce nitrogen, nitrate or sulfate to use in the cell. So the photosynthesis is divided two parts, but I'm focusing on the CO2 availability because CO2 is right now increasing in our atmosphere. We are facing to the high carbon period. But before the industrial revolution, CO2 concentration was almost 250 ppm. Right now, over 400. It's almost doubling. The CO2 is thought as a global warming factors, one of the big problem for our future. But for organisms, photosynthetic organisms, they need CO2 as a substrate. When they need they are eating the CO2 by photosynthetic carbon reaction is occurred in chloroplasts mainly. Of course, some bacteria originally green lineage come from the cyanobacteria to chloroplast in evolution. Carbon dioxide is the main supporter for human life. So, of course, many plants fix CO2, but predominant carbon fixation is occurred in ocean. Actually, you see how CO2 can be moved in the air. CO2 is very easily moved in air, but in aquatic condition, it's quite difficult to. CO2 diffusion rate is very limited in aquatic condition, over 10,000 times lower than that in the atmosphere. So the CO2 is not so available in water, and also under neutral or alkaline condition, CO2 is not predominant. So can you write down the chemical reaction between CO2 and water? If we have paper, we write down the chemical reaction. It is two-step. The CO2 and H2O react each other. The two different ions is produced in water aquatic condition. So, of course, this is on the slide, but the next step is one more step is occurring in alkaline condition, not seen in the natural condition, but carbonate is rare in the water because as the CO2 is much more predominant in acidic condition. If you can imagine the Coca-Cola, if you drink Coca-Cola with lemon, happens something in your mouth. Or some lemon in your mouth and drink the Coca-Cola. It's a big burst of the CO2 is generated from the bicarbonate ion. Therefore, the molecular species of carbon, we call it inorganic carbon. CO2, bicarbonate, carbonate, three different carbon we call inorganic carbon because organic carbon is many sugars or many, of course, amino acids also contain carbon. So before fixation, the inorganic carbon is molecular species of the carbon is important for photosynthesis. This is why the carbon molecular species important is enzymatic reason of the substrate specificity of the CO2 carbon fixation enzyme. Do you know the enzyme? RUBP carboxylase, oxygenase. It doesn't react with bicarbonate or carbonate. It only reacts with CO2 molecule. So the molecular species is very important. So I'm focusing, one is a diffusion rate problem and molecular species problem. Bicarbonate is dominant in seawater, but bicarbonate is not fixed directly by Lubisco enzyme. So they need much more CO2 inside the chloroplast because Lubisco is located in chloroplasts, and main molecular species in ocean is bicarbonate. So we call this kind of supporting photosynthesis supporting system we call CO2 concentrating mechanism. Of course, maybe this physiological event is found almost 40 years ago, but much more previously, of course, many other reaction was identified. But this is in your textbook, plant physiology textbook, you see that this kind of similar graph. You see the C3 plant like black pine, the C4 plant like corn or sugarcane perform much more better photosynthesis in high temperature. But in this case, the high CO2 grown cell, we call high CO2 grown cell mean the cells grown under sufficient CO2 bubbling. 5% CO2 is supplied. They perform this kind of physiological response against the concentration of the inorganic carbon, supplied carbon. But if when you culture the cell under CO2 limiting condition, we call low CO2. This is air condition, bubbling with air containing 4.04% CO2. They perform much better. This kind of physiological difference is observed just after changing the CO2 concentration, just after 20 minutes like that. So the aquatic organism can acclimate CO2 limiting stress condition. This graph is very important to think about what's happening in the cell, because cell can detect how much CO2 available. Because if the CO2 is much high level of CO2 is supplied, they don't need this kind of CO2 concentrating mechanism. They lose the CO2, CCM, we call CO2 concentrating mechanism. They don't have to pay their energy to build such kind of system. But in the CO2 limiting condition and with light to high light condition, they need much more CO2. But if the CO2 is not available, they need to take up and perform if you culture low level of carbon, so they can perform much better photosynthesis. So this kind of we call acclimation to environmental stress. This is we call CO2 low CO2 acclimation. So please reminder, this graph looks like an enzymatic reaction. We call we assume the cell as an enzyme itself, and Vmax like this, and K half. We you know the Kn value, but we call the K half because the cell is not the enzyme itself. We assume the cell as an enzyme, and if you calculate K half, half of the maximum concentration of the substrate of the half of the Vmax photosynthesis max value is if we culture the cell high CO2 condition, K half value is high, and if we culture the cell under CO2 limiting condition, the K half value is very low. That means very good for photosynthesis acclimation. So if the K half value is low, we assume the better photosynthesis performance. So next slide show you the different plant photosynthesis carbon fixation pathway. No, many crop plants, rice or beans, contain only C3 pathway, only carbon cycle. They don't have any this kind of CO2 supporting supplying system. In case of sugarcane or maize, they have C4 pathway, different cell type, and fixed carbon as a malate or like that. So using high energy, high light energy to make such kind of organic carbon, and they calculate in the around the carbon cycle to elevate the CO2 level around the Rubisco enzyme. Therefore, the C4 plants need much more high energy, but they can perform much better photosynthesis. If you culture maize under very low light condition, you cannot grow. We cannot harvest. They need high light, high energy, and also the high temperature they need. And another type of can in desert area, they have time clock dependent CO2 accumulation system. And also this two system is called biochemical system, we call. And in case of an aquatic organism, they don't have this kind of cell differentiation. Of course, they live sometimes single cell and only have a carbon cycle, but the biophysical system is operating. That means the bicarbonate transporters is very important. Therefore, I'm focusing bicarbonate transporter expression or like that. So in these days, you may know the Bill Gates Foundation support this kind of biophysical system can be transferred to crop plants to elevate the productivity. This is a very huge project in US and in England, and it's quite amazing. Our gene was used for transfer to the rice or many other crop plants. And Actually, this is model of the chlamydomonas cells. At first, I have to mention about the specialized localization of the Rubisco accumulated area we call pyrenoid, which is very rich in Rubisco enzyme in the chloroplast. The cytochrome membrane is penetrated into the pyrenoid to support the supply of bicarbonate or CO2 from outside. From stroma into the pyrenoid region inside, they need this kind of mechanism. And I'm just now focusing on the from outside, bicarbonate can take up by ABC transporter. And then from cytosol to the chloroplast stroma, another ion channel, LTIO named, is operating. And in this cartoon, I hidden one of the component in yellow color. Please write down the name of the enzyme. This enzyme is very ubiquitous. Several other molecular marker also in your blood. A lot of the enzyme you have. Kind of enzyme which react, which facilitate from CO2 to bicarbonate or bicarbonate from bicarbonate to CO2. So this kind of reaction is rate limiting. Therefore, initially this reaction is most important factor which limit the photosynthesis. And low CO2 condition, the enzymatic activity is elevated by changing CO2 from high to low. So my previous boss in Tokyo University published a paper. This enzyme is most predominant and important enzyme. So he worked with Benson, Calvin Benson. You know, the Benson supported his work. Do you know the name of the enzyme? It's very, very basic biochemistry. Anyone? No? How? First time to hear that? How about in NTU? Is there anyone who can answer the name of the reaction? Name of the enzyme? Which enzyme? In your blood, huge amount of enzyme. In blood cell. In your blood cell, a lot of enzyme. Yeah, if you make to move your muscle, your after production of CO2 should be evacuated from your body. Otherwise, your blood is becoming pH drop down. And this enzyme facilitates to convert CO2 to bicarbonate and transfer into your lung. And then the lung, in your lung, the bicarbonate is converted to CO2 and you breathe out CO2. It looks like almost 5% or 2% or huge high level of CO2 if every time you are producing CO2 after eating. So this enzyme, this enzyme is on the list, but you cannot see it. Carbon hydrase, which react, which facilitate exchange between the CO2 and bicarbonate, which the enzyme contains zinc ion, which is the center of the enzymatic reaction. Zinc is binding to CO2 molecule itself, or bicarbonate. The mechanism is very basic, although I don't touch in detail. Anyway, so this slide shows you the list of the low CO2 inducible proteins in graminomonas. So after culturing the cell under high 5% and transfer to air condition, they roots, they cannot acquire not so much CO2, so they need to develop CCM component. Therefore, we listed the protein gene product by comparing RNA by using RNA-seq analysis, transcriptome analysis, and just isolate the RNA from different conditions, from high CO2 cell and from low CO2 cell, and compare the expression level. And we listed from top to the, we listed almost 20 proteins, and we used the almost the top line. We named LCI-A, low CO2 inducible protein A. This is by chance my student named LCI-A, A, B, C, D, E, F, and some other group named LCI 1, 2, 3, 4, 5, 6. So a little bit confused, but anyway, LCI-A is very important for CCM, which is located in corpus envelope. It looks like it is already analyzed in E. coli, formate nitrile transporters family. It looks like in the membrane. And also the other protein, HLA3, which looks like HLA3 means the highlight activated gene 3. This was reported previously by Carnegie group. Highlight condition, they need this protein to survive. So it's structurally similar to many ABC proteins in human. So, sorry. Anyway, I can skip this field, but during the isolation of the CO2 requiring mutant, we isolated one mutant which cannot acclimate to low CO2, which doesn't express any of these all genes. Not all, but almost all genes was defective by missing CCM1 protein. This protein also contains zinc ion and looks like a CO2 sensor. We assume this could be sensing protein, but anyway, the carbon anhydrase, SCIA, ion channel, bicarbonate transporter, and other component is missing in this mutant. So these all CO2 inducible protein is regulated by single gene product. And this is a time course experiment by using the Western blot analysis. You will see the 5% condition, the no expression of the HLA3 and LCIA. Also, we named the LCI1, which is also plasma membrane protein. But as a control, histone protein is constitutively expressed. So after 1 hour later, faint band appeared, and within 2 hours, protein is induced accumulated in the cell. So this kind of reaction is totally dependent on the lowering CO2 in the presence of light. Without light, they don't need CCM. And the protein localization, what kind of method is available? If you identify new gene product, to assume the function, you need to localize the protein where it is, so that you can generate, for example, antibody by using rabbit or like that. Right now, previously I'm taking care of rabbit by myself, but right now it's very easy to just send the information of the gene sequence and you will get the antibody. Of course, you have to pay, but you don't have to take care of rabbit like that. And this is histochemical. Using the antibody, you will see the subcellular localization. The upper panel is HLA3. It looks like almost a surrounding area. It's also clear. And in case of LCIA, the shape of the corpus is very clearly indicated. That means the corpus envelope protein. LCIA is corpus envelope. And HLA3 is one is a histochemical analysis by using antibody, but another method is biochemical fractionation. You can isolate chloroplast from the cell and separate from the total protein, and you can compare the Western blot total protein and plasma membrane fraction or chloroplast envelope fraction. So the HLA3 is highly accumulated in plasma membrane fraction. And in case of SLA, chloroplast envelope fraction is very rich in the protein. So this kind of analysis is biochemical analysis. Tell you where it is so that you can make a model. I already show you the model. Initially, from outside, HLA3 is operating the plasma membrane and take up bicarbonate inside. So what is ABC protein? Can you write down the abbreviation? What is ABC? ABC is three initial alphabet, of course. A means the ATP, B is binding, C is cassette. My friend found the MDR protein from human in NIH many years ago. He has struggled in for a long time after joining the Arapasan laboratory in NIH, and he firstly identified MDR protein, and he succeeded to crystallize in these days. This cartoon shows you this is a ABC protein from algae, which is different from granulars, but this is high temperature resistant red algae. They don't know the substrate, but the protein is very, very stable. Therefore, they have succeeded to crystallize, and they showed us the model of how they work, and the structure is flexible by consuming ATP energy to in case of the MDR, which from the human cell is export of the drug, anti-cancer drug outside. So the main problem for cancer-resistant problem is occurred by expressing the MDR protein, which is export chemical outside the cell. So cancer cannot die. To avoid the chemical attack. In this case, or in Crummy case, bicarbonate should be taken up from outside to inside. The orientation is opposite. So you remind this kind of structural movement is dependent on totally ATP energy, and they support the CCM. So in summarize, one is many different components is essential to support CCM to CO2 concentrated mechanism. One is carbon carbohydrates, another membranes associated transporters, and other things is a Rubisco associated protein, which organize pyrenoid. And also the regulatory proteins is very important to switch on the CCM protein expression. Of course, the CO2 sensoring system is also operating in not know so deeply, but this is a problem for future. So this is just an introduction, and the Dr. Wansan started to find a new component of the CCM. So how can you identify new components of CCM? And different approaches are available. He started genetically. What kind of a genetic tool is available? Of course, transformation of the cell can be achieved in chromosomes. Isolation of the mutant can be achieved. So, of course, I talk about the HL transporters. We not isolated look at mutant from the insertion mutant library. Therefore, it's very good to isolate mutant which cannot survive under low CO2. So they need some component to support their life in low CO2. So this kind of selection is very clear to find out the relationship gene product and physiological function. So I recommend him to do this kind of things. Of course, many biochemical analysis by using previous non-carbon carbohydrates associated to protein or like that can be achieved. Of course, physiologically, you can measure different enzymatic activity, or at least this approach is very, very limited. So I prefer genetic approach to say the concrete result. This is my preference. So he made a lot of insertion mutants, 30,000 by himself. One by one, pick up the transformed colony into the microtiter plates. How many plates you can assume? Actually, after making this kind of library, during making library, he replica plated, double the plate, left side high CO2, right side low CO2, and compared growth in the different chamber by just changing the CO2 level. So the normal cell can grow high CO2 also under low CO2 because they can induce a CCM to support CO2 fixation. But some mutant which cannot induce CCM cannot grow under low CO2 condition. This is a good example, very, very nice mutant he found by chance. Of course, initially he said to me, 200 mutants I isolated, he said to me, oh, it's really good mutant. So I recommended him to focus single mutant. Very clear phenotype is very important. But this mutant H82 is very clear phenotype, which can grow high CO2 and the high CO2 but not so good under low CO2. So the long story in short, the insertion hydrolyzing concept is inserted with Cas protein gene. Cas is reported previously in higher plant. This is the Cas protein is. This mutant cannot accumulate in carbon. Also, wild type accumulate, but only half of the level of the accumulation is performed. So the CO2 fixation is affected by the lower take-up rate. This measurement is by using the carbon-13 bicarbonate, how the carbon is accumulated, incorporated into the cell. So you will see the 20 seconds. You can imagine the experiment after injecting the bicarbonate and illuminate 20 seconds, you have to kill the cell and fix and measure the carbon inside the cell. This is a very nice trick. We call the silicon centrifugation method. I'm not touching in detail, but the experiment is very hard. 20 seconds, 40 seconds, 80 seconds. So by using the different samples. And the K half, can you remember the K half? If the K half is low, is photosynthesis very good. Low level carbon is very nicely like a Km, low Km, that affinity is very high. The high K half means the not so good performance. So we already hit complement mutation by using Cas gene, intact Cas gene into the mutant. He succeeded to complement mutation. The phenotype is very clear. So this is very, some mutant we cannot complement. So we have to discard the mutant. So this kind of step is sometimes laborious. But if you face this kind of mutant, single insertion event is very important. If you see the different multiple insertion of the multiple mutation, you cannot get the complementation. It's quite difficult to answer which gene is important to your related to your phenotype. So the mutation was completely complemented by Cas gene. So this is a Western blot analysis. Almost many CO2 invisible genes were induced like wild type CCM1. This is a regulator constituted about CH1 carbon carbohydrates. ACI1 with ACIBC is induced also in this mutant. So not all the CO2 regulated gene is affected. Only two gene products, HLA3 and LCIA, both is missing in this mutant. And in the complemented line, the expression is completely recovered. It's very clear that this gene is transacting to the transporter gene regulation. It looks like a regulatory mutant because HLA3 and LCIA is normal, but no expression in this mutant. Therefore, the Cas gene product is regulating to transporters at least. So the Cas is already published in the Arabidopsis, which related with stomata opening and closing, and also the infection related. The Cas mutant in Arabidopsis is weak to the fragile infection. case of the green algae, you will see the right side. Acclimation to high-rise stress, that means it looks like low CO2 stress, because under high-light condition, the cell consumed out all the CO2. That means the low CO2 stress. And also the other group in Germany, they support the cyclic electron transfer in photosystem is affected. This is a very big argument between us and the cyclic electron flow is needed to support the CCM itself. Or this kind of support is reported inside bacteria previously, but still we don't know exactly. Anyway, so this is biochemical experiment which cancer is reported to guide calcium in the chloroplast. Initially reported as a proper plasma membrane protein, but in science, but as a group reported, this is really chloroplast thyroid protein. This is, the science paper is, I don't know what happened. But the first report of the cancer in apoptosis reported it was plasma membrane protein, but it's changed. And by using different antibodies, specific N-terminal or C-terminal, we isolate the thyroid membrane from cell and treat protease and to find out how the protein is located on the thyroid membrane. This is Cas protein contain one single membrane-associated region, but looks like soluble protein. So by using the control enzyme CH3, which is chloroplast cytochrome protein inside the cytochrome bag, this is protected by the membrane, but the Cas protein is degraded by treatment of the protease. That means that this Cas protein is located outside the cytochrome membrane. One other thing is calcium is binding. I skipped the biochemical experiment, but the protein is specifically bound to calcium, not to zinc, not to manganese or iron or like that. So we are seeing the calcium itself is affect the photosynthetic performance or expression of these two transporters. So by using the chemical which chelate calcium to reduce the calcium concentration outside the cell. So if you add a bacter, which bacter chelator of the calcium, the expression of the two transporters is strongly affected, and we can cancel by adding extra calcium chloride. You will see the recovery of the expression of the two proteins. Therefore, the calcium concentration is very, very important to regulate expression of two transporters. So in conclusion, the Cas protein is necessary to induce the CCM, especially the LCI and HLS3 two transporters are regulated by Cas protein. And also the calcium itself, concentration is very important. cancer is located in next talk, Shimamura-kun will talk about the details, the subcellular localization of the cancer protein. But cancer protein is located on the cytokine membrane outside, but not all the chloroplast location. He will talk about that. He will introduce a new paper published this year. Cancer is exposed to the stroma site. That means it looks like from the stroma, some signal should be detected, and the expression of the transporter in nuclear gene. It's very, very curious why the chloroplast and nuclear, two different subcellular organelles communicate each other. So that if you lose the cancer protein, the nuclear transporter genes are not expressed. We can assume that this kind of communication we call the retrograde signaling. Opposite word is enter signaling. Anterograde signaling. Many higher plants, chloroplast system is regulated by nuclear genes. Many, for example, Rupisco subunits come from nuclear with transit peptide. Almost all the chloroplast protein complex is made by chloroplast gene product with nuclear gene product. It's a hybrid. But in this case, cancer protein localized in the cytokine membrane regulate the nuclear gene expression. We call this kind of physiological, the retrograde signal is operating, which regulates the nuclear gene. So, do you have any idea what kind of signal is transferred from chloroplast to nuclear? If you are familiar with, I'm not sure, but in animal case, from mitochondria to nuclear, many disease related with mitochondria-related disease affect nuclear gene expression also. So, retrograde signaling is one of the keywords today's keyword. Okay, it's almost time to move the Shimamura-kun in my laboratory will talk about the subcellular localization of cancer protein. Before going to the students' presentation, shall we have question time? Oh, yeah. If you have a question, please raise the question. Oh, hello. Yeah, any kind of question please write down the question in your sheet also. Yeah, Dr. Heyman.
I just have one question for clarification. You mentioned downregulation of HLA3 in the Cas mutant. Does that also affect mRNA levels for HLA3?
Oh, I skipped the figure, but mRNA level, initially HLA3 expression is increased, but the maintenance of the mRNA is decreased in the mutant. In wild type, mRNA accumulate both HLA3 and LCIA also express mRNA accumulated and proteins produced. But in this case, the Cas mutant cannot accumulate in later, no mRNA. I assume that because of many regulatory factors, I said previously CCN1 itself is an essential master regulator, and retrograde signaling could support sometimes support mRNA susceptibility, but we still don't know how the mechanism biochemically. Yeah. Any other question?
Yeah, not directly related to your talk, but
sensing system is connected to cellular movement because the carbodominants can move. Yeah. So if there is any CO2 gradient, can they move to high CO2 place? They like oxygen, also the CO2. Because if you culture the standing culture, they go up to the surface to catch much more high light, much more oxygen, or much more CO2. In some experiment, light-dependent moving is already shown photoreceptor, and CO2 response is already reported, but we don't know the mutant. We really don't know we have never checked about this kind of mutant. They can go to the high CO2, like a mosquito. They want to CO2, so I assume this kind of movement may be affected. It's a good question. So, thank you.
Okay, let's take a 5 minutes break before the student presentation.
Okay, so shall we get started?
大好きしめな大好きしめなI will introduce high distribution subliminal localization of calcium binding protein Cas, a novel regulator of CO2 concentrating mechanism. Photosynthetic organisms can sense and respond to changes of several environmental factors such as light, CO2, temperature, and various nutrient availabilities to optimize or maintain their photosynthetic activities. Among these stresses, the shortage of CO2 supply impacts many physiological aspects of the plant in aquatic environments. CO2 linking stress is caused by the P7 force or diffusion rate of CO2 in aquatic conditions than that in atmospheric conditions. To acclimate to these stresses, many aquatic organisms possess a CO2 concentrating mechanism, CCM. CCM involves the active transport of inorganic carbon and enzymatic conversion between CO2 and bicarbonate to concentrate CO2. Molecular aspects of eukaryotic CCM have been mainly studied using the green algae Clendonis as a model. So far, it was reported that highlight activated C, HLA3, and low CO2 inducible protein A, ACIA, are associated with the bicarbonate transporting system. They facilitate bicarbonate uptake from outside of cells to the chloroplast across physiological barriers such as the plasmodium and chloroplast envelope. HLC belongs to the ATP binding cassette type transporter, and ACIA belongs to the formate membrane transporter family, and localizes to the chloroplast envelope. HLC and ACIA are cognitively associated with bicarbonate transport to increase the bicarbonate pool in the chloroplast stroma. These lossy-inducible proteins are related by zinc-containing regulator protein of CCM1. Recently, we isolated a novel high CO2 requiring mutant, H82. In H82, APH7 gene cassette inserted in gene of calcium binding protein, Cas gene. Next, although chondrous cells of wild type can survive in lossy conditions, in H82 cells, its growth rate was decreased in lossy conditions, and contributed strain C1 can survive in lossy conditions. In H82 cells, the accumulation of HLA3 and ACIA was decreased. In CL cells, the accumulation of these proteins are recovered. We believe Cas is essential for the operation of the CCM by blocking the expression of ACIA and HLA3. By proteomic analysis and indirect fluorescence assays, Clendonis Cas was also localized to the charcoal membrane and especially inside the pyramid. Pyramid is a prominent structure in the chloroplast of the cells cultured in low-C conditions. In many algae, the pyramid is surrounded with starch sheath in the chloroplast. Some bacterial membrane penetrates into the pyramid. It is pyramid tube. Glandular CAS regulates nuclear encoded genes related to the CCM, such as HLA3 and CRA. CAS-mediated retrograde signaling systems from chloroplast to the nucleus. However, the actual functions of CAS and its detailed subcellular localization in vivo remain to be determined. In this study, we reviewed distinct localization patterns of CAS in high-C and low-C conditions at high resolutions. This result could help in understanding the function of CAS associated with the retrograde signals relating stress-responsive genes. To examine the subcellular localization of CAS in vivo, we generated transgenic lines expecting CAS-fused clover. Clover is a clonidinous adapted modified green fluorescence protein. This plasmid was used to transform the H82 mutants, and non-transformers was designated as CL1 to CL9. These strains showed fluorescence signals derived from CAS clover inside the pyramid. By Western blotting analysis using an CAS antibody, a band of approximately 63 kilodalton was detected. It was corresponding to the predicted size of the CAS clover fusion protein. Among these transforming CO2 showed the strongest fluorescence signals and was selected on further analysis. The values of maximum observable activity, Max and Q-half, was similar to those of wild type. This result indicated that decreased photosynthetic bicarbonate affinity of H82 cells was complemented by expressing the CAS clover. High-resolution fluorescence images of CO2 cells were obtained using the combination of a sensitive hybrid detector and a image decombination technique. In high-C conditions, the fluorescence signals were distributed across the entire chloroplast, and several puncture spots were observed. By defocusing on confocal images, fluorescence signals displayed a mesh-like structure, and part of the green signals of CAS clover discontinuously overlapped with red chlorophyll autofluorescence. Enlarged fluorescence images were obtained by defocusing the sample from -0.6 to 0.8 micrometer from the focal point. Silver is 400 millimeters. Considering that CAS was detected in the fraction enriched with the charcoal membrane, CAS could be not uniformly but discontinuously distributed on the charcoal membrane in high-C conditions. Next, when the cells were shifted from high-C to low-C condition, the fluorescence signals were detected inside the pyramid as a distinct fluid-like structures. When we shifted the focus along the Z-axis direction, the strong fluorescence spot was also observed in the lateral region of the chloroplast. These fluorescence images were also obtained by defocusing the sample from -0.6 to 0.8 micrometer from the focal point. This fluorescence spot was overlapped with the region of eye spot. The autofluorescence signals of eye spot were detected in wild-type cells, but the signal's intensity was significantly weaker than that of CO2 cells. So, it is indicated that the fluorescence signals of the eye spot region in CO2 cells were mostly derived from gas clover. By focusing with confocal images in the pyramidal region, the whole-like structure consisting of several fibers were clearly observed. By increasing the contrast of fluorescence, this fiber were observed. A thin fiber is observed. And they could be derived from the structure of the pyramid tube. In lossy condition, the whole-like structure had almost disappeared, and gas clover was localized to the center of the pyramid. Considering that part of the charcoal membrane pyramid tube penetrated into the pyramid and freezes at the center of the pyramid, forming a muted core, and that the location of gas was not associated with the monoprotein synthesis. Dispersed gas clover in the chloroplast in Hershey condition could move and gather into the pyramid along the charcoal membrane during CCM induction. In this study, we determined subnormal localization of gas based on fluorescence images of functional gas clover in vivo at high resolution. Gas show distinct different localization patterns between Hershey and lossy conditions. The spiral localization of gas clover in Hershey condition changed to whole-like structures in lossy condition. In particular, this whole-like localization of gas clover was clearly observed for the first time. Although the relocation of gas in the chloroplast and its importance for the regulation of the CCM has been proposed, it remains unclear how gas moves along charcoal membrane. One possible mechanism is post-translational modification. Other CCM-related proteins also changes their localization in response to CO2 availability, and another phosphorylation from CO2 availability is limiting, but changing its localization is distinctly different from the gas localization patterns. Another possible mechanism is the structural dynamics of charcoal membranes. A recent study reveals that both the structural stability and flexibility of charcoal membrane is essential for dynamic protein reorientations. It is possible that gas also moves along with the membrane dynamics. Although the directional movement of the charcoal membrane from the dispersed chloroplast region into the pyramid and vice versa is unknown. It has become clear that the pyramid is important not only for CO2 fixation, but also for the regulation of the CCM. So far, hundreds of proteins with unknown function have been identified in the purified pyramid, and there could be other CCM-related proteins that could relocate in the chloroplast in response to the CO2 availability, as reported previously. Further screening of mutants showing around the localization patterns of these proteins could lead to understanding the regulatory mechanism of cellular localization in response to environmental stresses. We think high resolution images described in this study could be useful for observing the cellular localization of proteins, especially for ones localized in small components such as the plasmid in single cell. Thank you.
Thank you for your presentation. And does anybody have any questions?
One question, thank you to say it.
So maybe I didn't understand. I just want to make it clear that. So is this two cell so different two cell, or I mean, did you see the protein move dynamically?
Yeah, it is. Different conditions, CO2 concentration. In high CO2 concentrations, the gas is dispersed in chloroplast, but ratio condition, the localization is changed into the plasmid in the same cells.
You changed the condition?
Yeah.
And you can see the fluorescence just moving?
Yes.
Oh, yeah. Great. Thank you.
Do you have any idea to explain why the protein can move inside the chloroplast on the cytokine membrane?
One idea is so I don't have any idea.
Our own hypothesis. Then you can work on that.
Just one possible mechanism is post-tensional modification. So the CO2 condition is changed to high C to low C. The gas is modified by post-tensional modifications, such as phosphorylations.
If its movement is dependent on energy, can you inhibit?
Pardon? Sorry.
Can you inhibit this movement by adding some reagent?
In cells, calcium is removed, the just the organization is stopped.
You mean the calcium itself is important to movement? It's because of this protein can bind to calcium's selectivity, but the movement itself needs some component for example, actin or some filament, because the spinal cord is membrane, lipid membrane. So the direction should be determined while the inside to the organoid. After culturing low CO2 cell to high CO2, the proteins move out from the paranoid. This is a very big change. The protein localization can be affected by only changing the CO2 level.
So I think this is where you're headed experimentally. What happens if you mutate the calcium binding pocket of the protein? Would it affect localization?
So I can understand the question. So please.
Maybe you can try to guess. I don't think you've done the experiment, but maybe you can guess. What would happen if you made a mutation in Cas that affected calcium binding? Would that affect localization?
I guess the localization of these patterns is changed. So they don't sensing the they come to be not sense the calcium signals. So the localization will be changed from these patterns.
One other experimental question that you don't have an answer for. Do you think this also involves membrane reorganization within the paranoid? There may be membrane-associated proteins that are involved.
Yeah. I don't know, but yeah, just the possibility is higher. Possibility is high, I think so.
Thank you very much. So I have some additional comment about this movement of the protein inside the corpus. So the Cas protein itself, the expression or accumulation level is not changed constitutively, but the location is quite drastically changed by only changing by CO2 level. And of course, they need the Cas protein needed to induce transporters. Therefore, this Cas protein is a key person which regulate the expression bicarbonate transporters to accumulate low CO2, because without Cas proteins they cannot grow under low CO2. So we only know the subcellular localization, but we have previously the one can try to isolate the protein which are associated with Cas protein by using LC mass spec analysis, by using a tag Cas protein to complement the mutant by similar way, not using the Cas clover, but for example, flag tag. And he tried to isolate the associated protein from complemented line to find out the new component associated protein. But he has only detected ribosomal protein or elongation factor like that. So that we still don't know what kind of protein is associated and what is player to drive the movement. And one most important thing, previously pyrenoid is thought as just a structure which accumulated Rubisco protein around the thylakoid membrane. The pyrenoid is developed from the thylakoid membrane initially, and Rubisco get together by some other protein complex component, and the pyrenoid is developed. And then the Cas protein is also associated inside the pyrenoid, especially in pyrenoid thylakoid tube, because the thylakoid lumen has energy, because a lot of protein inside. And he didn't show the connection between the outside the pyrenoid thylakoid membrane totally connected with outside pyrenoid tube inside the pyrenoid. So that we assume that bicarbonate also can be supplied through the luminal side, and the Cas protein can be one of the not the player as a transporter itself, but as to detect the CO2 level inside the pyrenoid like that. Because Rubisco is itself consumes CO2 inside the pyrenoid. We can assume CO2 level is very, very low inside the pyrenoid. So such kind of sensing mechanism could be related to calcium also. So this protein is very, very important also in higher plant. So another thing, another protein, LCI B and C, also move from outside in the chloroplast, similar, but not inside the pyrenoid, but surrounding the pyrenoid. Some other component also moving in the by lowering CO2, they very rapidly get together surrounding the pyrenoid. So recently the LCBC protein is shown to be the carbon anhydrase. So that protein also moved to the pyrenoid and to support CO2 leak, to avoid CO2 leakage from the pyrenoid, to resupply to the thylakoid lumen size. So we still don't know the biochemical mechanism, what is going on there. We know the subcellular localization. We can make a hypothesis, different hypothesis. Then you can use a different approach. So making your own idea is most important. How to screen the mutant. Yeah. Okay. So it's almost finished. Thank you.
Are there any questions from anything you cover?
No question.
No. So we are running out of time. So
Thank you very much, Dr. Fukuzawa.
Thank you.
Yeah.
bye now. So we'd like to close the class.
See you next week.